Hello and good afternoon, everyone. Thank you for joining us today. My name is Anas, and today we will be doing session 2 of 3 of the Only Cell webinar series on antibody identification. So this session will be led by Helen, and for those of you who don't know her, let me give you a brief introduction. Helen Thom is a registered biomedical scientist who began her career with NHS Blood and Transplant as a placement student back in 2012. And subsequent to that, she secured a trainee BMS role in the red cell immunohematology laboratory, specializing in transfusion science. And after working in RCI for 7 years, she moved into the PBM team. So that's the patient blood management team, where she worked on various education initiatives. And then she moved into a project management role in hospital customer services. Helen is now part of the Transfusion 2024 program, helping to develop collaborative models between NHSBT and hospital transfusion laboratories. She's also a member of the Institute of Biomedical Sciences Specialist Advisory Panel for Transfusion Science and is a strong advocate for biomedical sciences. We're very lucky to have her here with us today. And with that, a big thank you to Helen for joining us, and I'll hand over to her to get us started.
Thank you, Anas, and thanks everyone for joining. So this session, what we're going to do is, like I said, Anna said, it's part 2. So I'm just going to do a very quick recap of the last session, and then we've got some cases to go through. I did send the panels out, but I appreciate not everyone would have had an opportunity to do those, so I will have them on the screen. And then there's just some polls that we'll go through. So hopefully everything will go nice and smoothly for you. And there's time for questions at the end as well. So this one is a follow-on from the last one. So this is about putting it into practice. So the last time we covered tools of the trade. So this is just what we're going to do today. So the last time I went over the basic principles of antibody ID, and I like to say, this is sort of bucket chemistry is probably the best description. So we're looking really a drop of red cells and a drop of plasma, and we use this. So you've either got unknown antibody with known antigen, which is how we'd determine what antibody IDs. We've got known antibody with unknown antigen, and this is what we use for regrouping and phenotyping. And ultimately, the antibody will bind with its corresponding antigen, whether that comes from the known or the unknown. We'll cover the indirect antiglobulin test. So basically saying when there's an IgG antibody present there, IgG isn't capable of bridging the gap between red cells. So you need to add a bridging antibody into that. So we discussed around how you use anti-human globulin for this. And what this does is it's a mixture of sort of IgG and complement, but it will bind your IgG together. When you centrifuge your samples, this is how you get agglutination when you're doing your IAT. Sorry, if you've got a problem with the sound, what I would suggest is just if you're able to leave and then rejoin, hopefully that'll sort it out for you. Then we'll discuss direct agglutination. So when you are using IgM, I've described it last time as a massive antibody, the best way to remember it. And IgM antibodies are capable of causing agglutination because they are capable of bridging the gap between the red cells, so therefore you don't need to use a bridging antibody when you're using IgM. So that's like your direct agglutination for your phenotyping, and if you were doing it like a direct spin or tube. So saying this is what you use for your blood grouping and IgM phenotyping anti-sera, and it can even be used to detect your cold antibodies, which is quite helpful when you're doing some of your investigations. Also discuss grading. So we said this is really helpful with your antibody ID because it can allow you to see if you've got a mixture of antibodies. So if there's multiple things going on, you might have different strength of reactions. So it's a really useful tool. It's also really useful for demonstrating dosage. When you've got antibodies, say an anti-M, for example, which if you missed a heterozygous line, for example, and it's only hitting the homozygous lines, grading is a really good way of using that. And it's also good for looking at your enhanced reactions, which helps you, if you're using papain, for example, that's a good way of sort of saying if a reaction is enhanced in papain, you would have a stronger reaction using your grading. Moving on to papain, so at the as you've probably discovered from the previous talk, I do like SpongeBob and co. So how I would describe PAP-beam treatment is when you are doing an IET, you've obviously got all of the antigens on the red cell, but to get those enhanced reactions, the best way to get an increased strength of reaction is to remove some of the antigens from the red cell. And this is what happens when you're patronizing your cells. You're actually getting rid of your Duffy and your M, N, and S's. So Patrick, if we think of him as Pappin, he comes along, treats the red cells with Pappin, and it's actually removing some of the antigens. So this is how you get those enhanced reactions with your RH, in particularly your kids as well. You can sometimes see it with KEL system antibodies, but sometimes the reaction can stay the same. But if you use an enzyme IET instead of just the enzyme nucleocards, you would generally see a stronger reaction. So that's how PAP-beam treatment works. There's no PAP. What I was saying as well is, so as I've just mentioned there, we've got the RH and KID are particularly enhanced. So if you think of Patrick's moving those trees out of the way, and actually it's just allowing a stronger reaction there, and Squidward can get a bit more of a suntan. I also talked about genotyping and phenotyping, so sort of comparing and contrasting the two almost. The phenotyping is performed using it's unknown patient's red cells with known antisera, whereas when you perform a genotyping, it's used with the patient's DNA, and this is extracted from the patient's white cells. So I've mentioned previously that if your patient has been transfused, you wouldn't be able to perform a phenotyping, but you can perform a genotype because of the way that we extract the DNA from the white cells. All the donor units are leukodepleted. Therefore, the only residual white cells would come from your patient. So phenotyping shows which antigens are expressed on the patient's red cells, and you can also use this for your paternal phenotyping, so that's a useful tool there. And the genotype shows which genes are present, but they may not necessarily be expressed on the red cell, but it does give you a predicted phenotype. Phenotyping can't be performed when the patient's been recently transfused, so we say within 3 months or less. And this is due to the dual population of cells, because if you were performing phenotyping, it's going to be a mixture of patient and donor cells, and you would get a false positive reaction or even a mixed field reaction, so you wouldn't actually be able to see whether the patient was antigen negative or not. Genotyping can be performed following a recent transfusion. And with your phenotyping, IgG positive DET can cause false positives with IgG antisera, so we did discuss that in quite a bit of depth the last time. But obviously you would be able to use this, you would be able to genotype these patients. And in genotyping, this can also be used to determine the genotype for hemoglobinopathy patients, and this is done at IBGRL. And it can also be used for fetal DNA as well, so it's a very useful tool. We discussed homozygous and heterozygous expression. So you can see this is an example that we covered in the last session. So if you have a homozygous cell, that is just where it's showing sort of single dose expression of one of the antigens, double dose expression of one of the antigens. So this patient's gene would have 2 copies of the GKA gene. Therefore, this cell is GKA positive, GKB negative. But if it's heterozygous, the person only has a single copy of each of those genes, so the GKA pos and GKB pos, and this also fits in with the dosage as well. We also discussed how antibody screening works, so I went through the IET process for antibody screening. We discussed the makeup of your panels and how this allows you to get your inclusions and your exclusions. So in the UK, following the BSH guidelines, when you identify an antibody, if it's a brand new antibody, you need 2 positive examples of that antibody that's negative for any other antibodies, and also 2 negative examples in order to see that that antibody is present. So we talked about that. We discussed the importance of understanding the clinical significance of antibodies, because sometimes, you may have low incidence antibodies that, while it could be a bit of a nuisance, they're not actually clinically significant and they're not going to cause your patient any harm. So you don't need to actively exclude those as part of your investigation. And we talked about an auto control as well, and how that would link when you're talking about your direct antiglobulin test. We also covered how to perform an elution. So I went through that hopefully to help you understand that when you are performing an elution, this is on a patient who has been recently transfused. The DAT is IgG positive and has been transfused within the last 6 weeks, or there's been a change to serology or a change in the strength of the DAT. That's the kind of time we'd be looking to perform elutions on certain patients. So now we're going to look at putting it into practice. So I appreciate that was just a very quick recap. I've got 4 cases to go through, because I'm terrible and I do talk rather a lot when I'm doing these talks. We'll see where we get with the first 3 and then go on if there is time, because I'd just like to leave some time at the end for questions if that's possible. So hopefully you won't mind if we end up having to skip the last one, because the point of these is, you know, to generate a bit of discussion as well. So the first case, so we've got a 75-year-old male, broken hip, they're in hospital, they've grouped as A positive, there's no record of any historical antibodies, the patient's never been transfused, and you've had a request from the ward that they want 2 units of red cells for this patient. So I'm going to give you a minute to have a sort of look at this. I do appreciate that it's probably not easy if you're doing it on the screen, but I don't know if anyone had an opportunity to kind of have a look at some of the panels. But I am just going to launch a poll. Hopefully it won't interfere too much with you looking at this screen, just to see what antibody you've identified. So I'm just going to launch that now. Fingers crossed something happened there.
Yep, the bill came up.
Lovely, thanks. I'm having that issue again, and I swear I can't see it.
Oh, no.
Got a couple of responses there.
So we're getting quite a few responses in the chat.
Oh, thanks. I have the chat open as well, and it's just not showing me. I don't know why. I want to go back to the polls there. So I can see I've got anti-N, which is quite large, so there's a few responses there, a couple of thumbs up to that. WB, LE, GK with a dosage effect, and an anti-M. So I've got 29 responses there. So I'm just going to close that poll. And so that was my slide that I should have put on. So what we'll have here is an anti-N. So hopefully you can see this here, and apologies if that wasn't enough time for everyone to get to that. But we do have an anti-N. It is showing dosage. So if you look at cell 2 there, that's a heterozygous line, and you do actually have a 1+ reaction, whereas on the homozygous cell lines, you have a 2+ reaction. So this just shows how that can actually show dosage. And this is quite common when you're looking, when you have anti-N, because we looked at M the last time, and that was a so-called antibody. They're in the same family and they behave in a very similar way. And it's also one of the extra, another thing to think about is when we're discussing the tools. So this one, you know, it's typical N, it's that classic showing dosage, but it's also destroyed by papain, and that comes to that understanding how antibodies behave, understanding your antibody interaction, and whether the how the impact of papain as well. So papain has shown us there that that's another way to confirm that it's there. In this case as well, we actually have sort of 2 positive and 2 negatives on that as well. So, and you can see this here in the inclusion. So you've got lines 3 and 6 there will give you your positive confirmation. There's actually lots on there, but we'll just use those 2 as examples. And when you're thinking about your exclusions, you can actually exclude everything else on that panel as well. So if you haven't had an opportunity to do the panels, if you want to do them later on and have a look, you can watch back the recording just to see what you're getting for exclusions on there. So for this patient, we would want to perform some phenotyping. The patient has not been transfused, so we're okay to do this, and would be using IgM antisera. And we've tested our patient, and the result is that they are N negative, so this would be an allo antibody. So the next question that I'm going to launch here for you is anti-N a clinically significant antibody? And there's just some different answers there. So the first one is all M&S antibodies are clinically significant. Allo-anti-N is only clinically significant if detected by IAT. Allo-anti-N is only clinically significant when detected by direct agglutination. Or allo-anti-N is not likely to be clinically significant. So if you could just pop your answers there, and I can see already that, so we've got 25% of people saying that all M&S allo-antibodies are clinically significant. Few there saying it's only clinically significant if it's detected by IAT. A lot of people are going with it's not likely to be clinically significant, and then there's a little handful of people there saying it's only clinically significant when detected by agglutination. So the winner so far is allo-anti-N is not likely to be clinically significant, and that is actually the correct response to that. But these talks, they're all about education and it's about learning, so there's no, you know, if you didn't know the answer to that, that is absolutely fine. That is what your guidelines are for. So for here, I have actually just incorporated some information around the whole M&S system. So M, N, and S, these are antibodies which are destroyed by papain except for anti-U, and they typically demonstrate dosage. Anti-M, so that was a little bit of a trick question. Anti-M is clinically significant if it's reactive by IAT. And for M, you would require antigen-negative red cells if it's detected by IAT. But for anti-N, if you look in the guidelines, it's unlikely to be clinically significant. So actually, for this antibody, you only need cross-match compatible by IAT. Anti-big S and anti-little s and anti-U are all clinically significant antibodies, and all of those require antigen-negative blood. So you can see, even within the system, you know, within the blood group system, I'm saying that these antibodies sort of behave the same way, but actually the clinical significance can change with these. So next question, what blood would you select for this patient? So I'm just going to launch the poll, if you give me a sec. I have lots of questions down the side here, sorry. So if I launch that for you now, hopefully that will appear for you. So what blood would you want to be, what would you want to select for this patient? So your ABO, D and K compatible, ABO, DK compatible, N-neg, ABO, DK compatible, extended RH matched, or ABO, D and K compatible, extended RH and N-neg. So got, so far 75% of the people are saying the first answer. Few have gone a bit rogue and are saying that we would be selecting N-neg for this patient. And I mean, if that's what you would do following your local policy, that would be okay. However, the correct answer is the top answer, which is ABO, D and K compatible, and that would be cross-match compatible by IAT, so you don't have to select antigen negative blood for the anti-N. I'm just going to close that poll there. So that brings us on to our next case. Just before I move on, does anybody have any questions? If you do, if you just want to pop your hand up or pop something in the chat. If not, I will move on to the next case. No, I can't see anything there. So the second case, we've got a 32-year-old female. They are pregnant. This is their booking bloods. They are 11 weeks gestation. They've been grouped previously. They are B pos, big E neg, K neg, and they are also Duffy B neg. So we have a historical phenotype on this patient. They have a previous allo-anti-Duffy B, which in their previous pregnancy was too weak to titrate. The patient has never been transfused and they've been referred to your laboratory for an antibody ID. So this is the case panel. So I'll just give you a few moments there to have a look at that, and I will launch the poll if you want to put in what you think the antibody is. It's just a word cloud, so we just want to type it in.
So while people are doing the poll, just a quick question that came up in the chat. So somebody's asked, what does cross-match compatible by IAT mean? It's a phrase that we use quite a lot in transfusion. They just want to know what it means.
Right. Cross-match compatible by IAT, so is when you test your patient's plasma against the red cell unit, the red cell donor unit that you have selected for that patient. So say, for example, the previous one we've just used there, so we're saying we'd want ABO, D and K compatible. So that would be matching the ABO and D and K, which is most likely probably be K negative for that patient. But cross-match compatible by IAT is when you then perform a cross-match. So because this patient has an antibody, you would be having to perform a physical cross-match. It's not suitable for electronic issue. So when you perform the IAT, so mixing those donor red cells with the patient plasma, you get a negative result. So what that means is you've done the cross-match and it's compatible, therefore it's negative, so it's appropriate to use that blood for that patient. So if you did if we cross-matched that previous patient with ABO, D and K compatible, and the unit was incompatible, we wouldn't select that unit for that patient because it's not cross-match compatible by IAT. I hope that makes sense. Thanks. So I shall move on to the next one. Unfortunately, my chat has decided to freeze, Anna, so I've got it open on a separate screen and it's still not shown, but I can see the responses for the poll there. So we have anti-Duffy B. Yes, that is correct. That is what is there. So I'll just close that poll. Thanks for answering that one, everybody. So we've got an anti-Duffy B, and if we're looking at we're sort of exclusions and we're inclusions here, you can see in this case there are some heterozygote lines on the panel, but you've got a 4+ reaction, so that's a really strong reaction, and it's even 4+ against the heterozygous lines, because sometimes with Duffy you can actually demonstrate dosage as well, but in this case it's looking like a really strong antibody. So it's a particularly strong example of that. And there's Patrick again. So Duffy antibodies are destroyed except for Duffy 3, but Duffy E and Duffy B are destroyed by enzyme. So that's just sort of a confirmation that it is a Duffy B that you are looking at. And we can get our exclusions there because I've gone through and just made sure that we can get rid of everything else as well. So remember this sort of confirming the antibody. So I was saying previously, if it's a new antibody, you need 2 positive examples and 2 negative examples. But if it's a known antibody, you only actually need 1 cell to confirm that the presence of it is still there. But we do actually have more than 1 on this one, but just as a note, if you were wanting to go there. So
Sorry, Helen, before you go on, if you go back to the previous slide, we've got a couple of questions about that. So so somebody commented Kel could be an antibody. Can we explain why it isn't in this case? And the second question was regarding the panel you sent has Duffy A negative in the second line.
It's Duffy A negative in this. Sorry, I'll go to the Kel one first. So with the Kel, so you've got line 2, it's 4+ by IAT, but there's nothing there by enzyme. So that would technically be masking the sort of big K. But if you look at line 6, that is big K positive, and you've got you can exclude it on line 6 because that's negative by IAT. So you would be able to exclude the presence of big K using line 6. I hope that helps.
Yeah, that's perfect. I think the issue is the panel sheets that we sent out via email, they're slightly different to this panel sheet. So on the panel sheet, it had cell 2 as being negative on Duffy A, hence why there was a bit of confusion. All
right, I'm very sorry about that. I must have thought I'd copy and pasted this one. Perhaps I've copy and pasted another one, but that's my error. Sorry, I do apologise about that. But yeah, so was it still Duffy and egg? It just wasn't showing heterozygosity. It was just positive for Duffy B still on that one.
Yeah, I think some people have commented Duffy B as well.
Yeah, I'm very sorry about that. So yes, that's my error. I do apologise. I will send correct ones out at the end if you want use those. I do apologise about that. So hopefully we're okay to move on there. So with your Duffy, it's destroyed by PAPI except your Duffy 3, and it can show dosage. It is clinically significant. Duffy A and Duffy B are both clinically significant, and you would need antigen negative red cells. Anti-Duffy 3 is a high incidence antibody, which is also clinically significant, and you would need antigen negative red cell for that, which would be Duffy E neg, Duffy B neg. And it's destroyed by PAPI. So for this patient is actually pregnant. So what you would be wanting to do at this point is titrate the antibody, because this is a Buchan sample. So it was 2 weeks to titrate in a previous pregnancy, but this is, we're starting from scratch with this pregnancy, and you've got a really strong example of the antibody there. So I just wanted to show how you would perform your titration. So what you use is doubling dilution. So you can see there I've got on the left-hand side, that is the patient's plasma. So you would take 200 microliters of the patient's plasma, and each of these subsequent tubes contains 200 microliters of PBS. So what we'd do with these is take 200 microliters of the patient's plasma, put it into the first tube with your PBS, mix it up, change the pipette tip so that you don't get any carryover. Then take 200 microliters of that slightly diluted one, move into your next tube, change the tip, move on, so on and so forth. And this is how you do your doubling dilutions when you are doing titrations. So ultimately you are diluting the antibody with PBS. So by the time you get to the last tube, you've got your dilutions for your titration. Then you test these against heterozygous stuff. So that would be Duffy A pos, Duffy B pos cells, which I'll show you on here. The reason that you titrate against heterozygous cells is so that you can detect weak expression. So I saying when a cell is showing dosage, it will show a weaker expression. In this case, we want to look at the weakest expression that we can possibly detect. But for this one, and these are an actual photo of a case from when I was in the laboratory. This is a really strong, you can see, you know, in your neat dilution, you've got that 4+ reaction. Even in the 2 and the 4, you've still got a 4+ reaction. Even in the 8, so that's, you know, when you've diluted it, a titer of 1 in 8. When you get into 16, it's starting to weaken there. Up at 32, it's still quite a strong example of antibody. But if you look at 64, I would say we've got a good 2+ there, and there's a slight fleck on your 128. So when you're performing titrations, what you do is you report the last positive unequivocal pos that you can have. So you would be seeing, in this case, I would say the titer is 128, because there is still a positive reaction. Even though it's a very weak reaction, it's still positive. So I just wanted to put that in just so you can see how we actually would perform a titration. So with that, I'm just going to launch the next question, and we are just going to ask, is this a clinically significant titer? So yes, it's clinically significant because it's a clinically significant antibody and the titer is above 32. Yes, this is a clinically significant antibody and the titer is less than 32. Or no, this antibody is not clinically significant and you didn't actually need to titrate it. So I'll just let you sort of pop that in, see what people are saying around that one. And as mine hasn't changed, or it has there, sorry, I was going to say, is anyone putting any? Sorry, I've just had to do a little refresh there because my computer is not playing ball today. So we have 95% of respondents are saying that, yes, this is a clinically significant antibody and the titer is equal to or greater than 32, and that is correct. So if you did have an antibody and it's been titrated, you would titrate your clinically significant antibodies. So in this case, we want to see what that is. And when you look at your BSH guidelines, for Duffy, a titer of equal to or greater than 32 is a clinically significant antibody. So therefore, in this pregnancy, there is a risk of HDFN because that is a really high titer. So this one's just a word cloud that I'm going to launch. So I just want you to think about, is there anything else that you would want to consider? What would be your next steps in sort of working out what the risk is of HDFN? We know that the mum is Duffy B neg. We don't know if the baby is Duffy B positive or Duffy B negative. So yeah, so someone's put their partner test in. That's perfect. Just wondering if there's anything else. I think that's quite big there. There's a few people have sort of put in that. Yeah. Tested dad, cell-free fetal DNA. Yeah. Paternal testing, paternal phenotype. Yeah. Yeah. The biological partner. Excellent. Great answers, everybody. Thank you. I'm just going to close that there. So yes, paternal phenotyping, or you could do paternal. You could do paternal phenotyping or fetal genotyping. With Duffy A and Duffy B, we don't actually use the cell-free fetal DNA for that. Not currently. What you can do though is you can get a fetal genotype for this, using your chorionic villus sampling. So you can use that, or you could amniotic fluid. So it is possible to get a fetal genotype. You just wouldn't be able to do it from cell-free fetal DNA. Other antibodies though, such as D, G, big C, K, you can use cell-free fetal DNA for that. But really good answers, thank you. So when you're doing your paternal phenotyping, so hopefully you'd be able to get a paternal sample if you weren't going down the genotyping option, that is. So we want to test the paternal Duffy B phenotype. So is the partner Duffy B negative or Duffy B positive? So this is what it would look like if the paternal sample was Duffy B negative. This would be the offspring because both parents are Duffy B negative. All of the children would therefore be Duffy B negative. So in this pregnancy, if the paternal phenotype was showing that it's Duffy B negative, we would be seeing, even though the titer is really high, the baby is going to be Duffy B negative. Obviously, you always have to sort of consider that the paternal phenotype may not be the actual paternal person. So, but that's something for a whole other conversation. But if we did paternal phenotyping and the partner is Duffy B positive, it doesn't necessarily mean that all of the offspring would be Duffy B positive, because that's going to depend on the heterozygosity or homozygosity of the Duffy B. So you also, in this case, would be wanting to do the Duffy B, the Duffy A phenotyping at the same time. And your options are, if the partner phenotype, or if they were heterozygous, you're going to have a sort of 50% chance that the baby is actually going to be Duffy B positive. But if they are homozygous, so the partner is Duffy A neg, Duffy B pos, all of the offspring would be Duffy B positive. So depending on the sort of antithetical phenotyping of the partner, that's how you can use phenotyping, which I'd said earlier on that is actually useful in partner phenotyping. This is how we would do it in the laboratory. Or you've got that option to do the fetal genotyping. So well, that's that second case done now. So are there any questions before we go on? And again, please accept my apologies for sending out the incorrect panel. I do have a lot of panels at my disposal. I've obviously just pressed the wrong one when I've sent them over.
No worries. We've got a question in the chat regarding the phenotyping of the father. So if the baby was conceived through a sperm donor, how would you go about doing the testing then?
I don't actually know how that would work. I don't know. It's not something I have ever experienced. I don't know if that was something you would have to speak to the clinic that they were going to, and if they would be able to obtain a sample from the sperm donor, because it's a really good question actually. I suppose it would depend, you know, if that person, if they'd shared their information and were happy to have further testing done. I don't know, you could probably ask them, but certainly I'm going to go away and have a look at that because I think that's a really interesting question. But I think it would be depending on if it was anonymous or not. Someone has the hand up, they may know the answer, which will be quite good. Thanks.
Yep, so I've enabled the mic for them. So if you want to unmute yourself, you can ask a question.
Hi, my name is Regina. I'm from Kettering General. Just to answer that question, actually, especially if it's an IVF pregnancy, it is likely the clinic will be able to provide that information because of the comprehensive testing they have for the donor. So it is part of normally that information they give you in advance, much prior to even go through procedures, just to contribute to the answer.
Oh no, that's brilliant. Thank you so much for that. Thank you. Because like I said, I didn't actually know that. So it's really helpful that you've been able to come in and great question. Whoever asked that, thank you. Sorry, I can't see the chat to see who that was, but thanks so much for your contributions there.
No problem.
Right, we shall move on to case 3, and unlike me, we still actually have some time left, which is quite good. So here we've got a 65-year-old female. They're having a hysterectomy. They are all positive, no historical antibodies, never been transfused, and you received a request for an antibody ID. And this is your panel. Hopefully I sent you the right panel out for this one as well. So I will just launch the next poll. So this is just asking what you found. Yeah, so it's got a good one. They've gone anti-little c, anti-big E, anti-big E and little c. Couple of little c's, and anti-little c big E compound. That's an interesting one. Oh, couple of little c's there. Good answers. Thank you, everybody. So I'll just close that poll there. Sorry for anyone who's not had a chance to answer there. So keep doing that. So what I've done here is I've just sort of highlighted what the panel's looking at, just to make it a bit easier for those who might not have had a chance to look at the panels previously. So you can see there, it is looking like there's an anti-little c there. And because we've got, if you look at lines 3 and 5, we've got a 4+ reaction in comparison to the 2+ reaction. Sorry, that 3+ reaction there from some of the other lines. There's a mixture of sort of 2+ and 3+. So line 4 there is 2+, because actually that could be little c showing dosage. So it's looking like there's an anti-C, and it also looks like there's an anti-big E, but we can't say for definite, because remember, you need those 2 positive examples and 2 negative examples, and the little c is currently masking big E, even though it kind of looks like it could be there. So I'm not saying that it's there, but I'm not saying that it's not there. We can get some exclusions on this panel, but we can't currently exclude anti-big S, and we can't exclude KPE. So the next question I'm going to ask you is anti-KPE clinically significant? So you've got a yes, no, or unsure. I'm just going to respond so I can see the polls. So I've got the majority of people saying there that it's not clinically significant, some that are unsure, and that's absolutely fine to be unsure. And I answered yes, just to mix up the poll a little bit. So I've got 32 responses, so I'm going to close that. So anti-KPE is one of those where it's unlikely to be clinically significant. But that would be in terms of a non-pregnant patient. But for this investigation, it's unlikely to be clinically significant and would be wanting to select that cross-match compatible by IAT. So next one, why can't you exclude anti-big S on this panel? So I've just copied the sort of cell lines over there. So is it because the big S cell is homozygous, or is it because the big S cell is heterozygous? So why can't you exclude that? So everyone so far saying it's because it's heterozygous. A little bit of a change there. Some saying the cell is homozygous. Give it another few seconds for people to put some responses in, and then we shall move on. So we've got 90% of people saying we can't exclude anti-big S because the big S pos cell is heterozygous, and that is the correct answer. So if you remember the last time we were saying, looking at your BSH guidelines, when you exclude PID antibodies, Duffy antibodies, and S antibodies, you need to have a cell which is homozygous expression, and this cell is heterozygous expression. Therefore, we can't exclude it. Use that one to get rid of anti-big S. So what you would have to do in that case is put up a homozygous big S, little c neg, big E neg cell. So you'd probably be able to get that on your second panel. So the next question that I'm going to launch is, how could we see? So it's looking there, it does really look like there's a big E present, but how can we actually see if it's there? So do we need to test against little c neg big E pos cells, little c pos big E neg cells, little c pos big E pos cells, or you can just tell it's there from the reaction strength. You don't need to do any further typing, or we could perform phenotyping and see if that would resolve it. So most people are saying that you can test against big C pos, big C, little c, I'm not having a good day, am I? Little c neg, big E pos. We'll have some people saying that you can perform phenotyping. You can absolutely perform phenotyping if that's what you wanted to do, because if the patient was big E positive, you may not necessarily need to go and exclude, and you wouldn't need to give antigen negative red cells. So thanks for responding to that. So I'm going to move on. So what I would have said for that one is really you want to be confirming the presence of it. So you're going to need to test against the top one, which would be your little c neg big E pos cells. So good luck finding those in a normal lab, because the majority of panel cells, the ones that you're using are going to be your little c pos and big E pos together. But remember previously, so I said we use for panel cells, and these are made up in a particular way. You've got your primary panel. You might have that in CellStab, or you could even have it in Elsevers if you do tube techniques in your laboratory. But and a lot of you will have your secondary panels as well. You've got your screening cells that you may be able to use if you wanted to do that. What RCI have is a rare cell panel where you can use that for your high incidence negative. But we'll have what we call an R1R1 panel, and this is a panel that we use when a patient has an antenatal C, because it lets us see what's underneath, what's happening, where it's masked on your normal panel, as you would see. And there's also an R2R2 panel, and we can use these when the patient has antenatal E. We also have DTT treated panel cells, which can be used for patients on monoclonal antibody therapy, and I'm going to cover that in session 3. So one of the best ways to understand it is, one of the things I would learn about is the RH nomenclature. So this is like your sort of your Fisher-Rees table, as you might call it, and I've very kindly pinched this from the BBTS book, Introduction to Transfusion Science. So having your phenotype, so if you've got a phenotype there, so the first one is D pos. That hasn't done it properly, sorry. Oh, I'll move on to this one because it hasn't done what I wanted it to do on the previous one. So you've got, if your R1R1 would be D pos, big C pos, big E neg, little c neg, little e positive. So what you do is use these combinations, and it just gives you a bit of shorthand. So if we're wanting to look at an R1R1 panel, we need a cell which would, when you're looking at your R1R1 panel, you actually have this. So it's very similar to your other panel in that it's made up of group O donors, but this time it has all of the phenotypes apart from one. All of the cells consist of the R1R1 phenotype. So there's only one cell, which is cell 5, which is little c positive. The rest of the cells are all little c negative. Therefore, because we have other cells in there, so we have little c negative, big E positive cells, when we're using that, that is how we would confirm the presence of a big E. So if you have a little c in your laboratory and you want to send that across to the reference lab, if they use this R1R1 panel, they could actually see if the big E is present or not. Someone did mention previously in the word cloud about compound antibodies, and I'll sort of, if I can, I'll try and briefly cover those the next time. But they can also be used to identify compound antibodies as well. So that is how we do that, and we can actually get rid of exclusions. So for this one, we can get rid of anti-big S because we'll have some homozygous little c neg, big E neg cells on there. And we can also exclude anti-KPE. Now with the R1R1 panel, what you'll notice there is it's not by enzyme. So we don't use this, it's not a patternized panel for this one. So we wouldn't get those enhanced reactions by enzyme because it's just an IAT test. So this is going back to a normal panel. So when you want to confirm the presence of your little c, you've got 2 positive examples and 2 negative examples. Notice I've used the ones that are the negative examples and positive the negative for the big E, and they don't contain big E. And we can use the R1R1 panel to confirm the presence of big E. So you've got your 2 positive examples, 2 negative examples, and they give you the exclusions for the other ones as well. So for your phenotyping, we need to know the little c, big E, and K phenotypes for this patient, and you are going to perform some extended RH phenotyping. The auto control's negative, and even though this uses IgM anti-sera, you'll remember the last time that I said just to consider the auto control. So we do some phenotyping here using direct coordination. You can see the positive reactions there and the negative reactions are on the bottom. And as always, you can include a reagent control in there. So your RH antibodies, they're enhanced by papain. You would match for extended RH once you've detected those antibodies. It's the most, anti-D is the most clinically significant antibody after ABO. It's the most immunogenic after an ABO. A lot of them are implicated in HDFN. So, you know, your Ds, your big C, little c, big E, little e, these are all clinically significant antibodies. You would need antigen neg for all of these. But there are some RH antibodies where the may or may not be clinically significant. So when you are doing any of the investigations, just make sure that you check your BSH guidelines to see whether they are. And what I've put in here is I have an example of a little E panel. I haven't done it as a case. I just wanted you to see, because I've done the R1R1 panel, so this is the R2R2 panel. But what you would notice about this, so this would use this if a patient had an anti-little E. So this is what your normal panel would look like. You only have one negative line on line 3. It can give you some of your exclusions if you are obviously wanting to, but it's missed quite a few things. It's not covering everything. You've only got that one line. And then if you can see line 3 there, I've shown one there that's heterozygous for your KID antibodies, so you wouldn't be able to exclude your KID antibodies using this panel. So what we do have is the R2R2 panel. So this would allow you to get all of your exclusions if the patient only had an anti-little E. But unlike the R1R1 panel, there is no positive little E positive cell on here. All of the cells are little E negative. So it's just understanding your phenotypes when you're sort of performing these investigations. But when you're doing your confirmations for your big E, because we've excluded the presence of everything else, you can go back to your first panel and see, right, I've got 2 positive examples on that one, and then you will have your 2 negatives on this one. So you've got to sort of use them in combination to confirm the presence of the antibody. But that's how we use those panels. Right, I'm gonna, if it's all right with everyone, what I'll do is I'll just skip past this last case, and I do apologise about that because I would have liked to have sort of shown you. Why is that not? Because I've got a question to ask, and then if we want to, we can always, if anyone else has got any questions. So sorry. I was hoping I could have just skipped past. So I have a question that I would like to ask the group. So the last time when we discussed, when we did the previous one, we discussed the tools of the trade. I had said at the end, if anyone has any questions that they'd like to submit. So this is the one. This is one of the questions that I got. So why do pregnant women get weaker or completely lose their reverse group? I see this a lot. So I have tried to launch a poll there for a word cloud, because I'll be honest, this is not something that I've ever experienced before. I did share it with some colleagues who hadn't seen this either. So I thought the best place to bring it is to a group of people who work in a laboratory, and this may be something that you have seen. So I'm sort of turning the tables here and asking for your expertise on this. So if anyone has experienced this, where they do the ABO on their pregnant patient and they've got no reverse group whatsoever, or it's much weaker. Has anybody ever seen that before? I just thought I would ask. And hopefully we can help get an answer to this question. Someone said not sure. Like I said, I didn't know. It's not something I've not seen. A few. No, not seen. No idea. That was me. It wasn't something I had seen before. I was almost thinking, is it some sort of dilution effect? Because I know you can get that with like Lewis antibodies. But I haven't seen it ever with ABO. So it doesn't look like anybody else has either on this call. So elderly, yeah, I've seen it with the elderly, but I've never seen it with pregnant patients before. Oh, I did have a Google. The classic source of information. I didn't go on Wikipedia though. But it wasn't something that I had seen. So like I said, I just thought I'd bring it to the group. Someone's put transfusion training there. So I don't know if that's, you've seen it in your transfusion training, or not sure. Yeah, it could be the methodology, couldn't it? You know, I think sometimes if it's like this, it might just even be worth repeating. The group would probably be my recommendation for that. Well, thank you very much, everybody. Yeah, medication, somebody's put there. Yeah, that's not something I had considered. Plasma cells, reagent issue, medication, yeah. Well, that's all good answers, thank you. I think it's something I need to go and probably do a bit of research on, although I did try to do a little bit ahead of the session, but just couldn't seem to come up with anything. So might even be that, I don't know, it's something very strange going on there. Right, so I do apologise for not having time to go through the final case. Oh, yeah, got a hand up there, Medina.
Hi, sorry, just how could we get both videos really? Because we've missed the first part and you covered quite a lot there. And here we're like a group in our hospital, and we really are quite keen to kind of go through the materials one more time, because the presentation was great. Thank you for that. It's just we want to get all the slides and everything. You said that it will be on YouTube as well, but I'm struggling to get even the previous one, and I've exchanged a few emails with people.
Okay, I know they are recorded, and I'll be able to give you the sort of details. I'm happy for anything that I've shared. So I know I had sent the cases out previously just as individual cases, and obviously like I said, I've must have done that copy and paste job incorrectly. But I'm happy for the slides that I've shared across my sessions to be shared with anyone who was registered for the group. So by all means, I'm happy to send those to you. What I could even do. Yeah, what I could even do is speak to Anas and even just record separately just the end session and get him to tag it on the last case and ask him to tag it on, just because I do apologise, but I tend to talk a bit too much while I'm doing them. So
also, how could we get the previous one? Because we did try to sign with a colleague of mine, but the link wasn't working, so we couldn't get into the first bit of those webinar series.
That's totally fine. I know there were some issues with people joining in the first session, so apologies for that. What I'll do is after this session, when I send out the certificate of attendance, I'll also send out the slides and links to the previous recording and also this recording, so that anyone who wants to catch up can do so.
Okay, that would be brilliant. Thank you very much.
Thank you. And thank you for your help with the answer earlier on as well. Right, we've got a couple of minutes if anyone else has any questions.
Sure. A few people have said case 4 is quite difficult, so it'd be good if you were able to explain it. So maybe what we can do, we can have a recording after this session, and I can share that along with the other recordings.
Yeah, no problem. Very quickly, I will explain. Well, I will do a quick recording with Anas of this afterwards and go through that. Again, apologies for not having the time to do it today. But it's an anti-F. So just have a little think about that. You might want to have a look at F, but anti-F, it looks very much like anti-little c. I'll just see if I can flip back to the slides where I show you what it is. So this is what an anti-F actually looks like. So very, very similar to an anti-little c, but anti-F is a compound antigen. So it's a little c and little e in combination on one gene. It makes it, because it's a compound antibody, just think of them as sort of like being glued together. So it's CE, it's not anti-little c and it's not anti-little e, it's anti-little c, little e compound. So that was a sort of a bit of a sneaky one. So it is a shame that I didn't have time to go over that, but I will send the slides out and we'll do the recording and tag it on to the end of the other one. So even if you don't watch the whole thing again, you can just skip to the end and hopefully I'll explain that quite well in the slides for you. Thank you.
Perfect. Thanks, Helen. I'm just going to take control. Once again, thank you, Helen, for a informative and interactive session. I think the antibody investigations and sending out the cases beforehand helps people to do the exclusions and also participate in the session. And also thank you to everyone that joined us today. So like I mentioned, I'll be sending out certificates of attendance over the next couple of days, along with details for the next session, which will be the final session in the series. And if you attended this session as part of a group, please send me an email with details of the individual attendees so I can make sure everyone receives the certificate. The email will also contain links to the previous recording, this recording, and also the slides, as Helen mentioned. And if you haven't already done so, I'd encourage you to register for the OnlyCells webinar mailing list. And that way, whenever I organize a webinar, you can get information directly to your inbox. And if you're on a desktop, you can click on the link on the screen, or you can scan the QR code. Thanks again, everyone, for being here, and we look forward to seeing you in the next one.
Thanks, everybody.